So I'm going to start off with just some mention and thanks to my collaborators on this project, people at Cortigen, Net, Christine, who runs our clinical group, and some of our bioinformatics folks who helped analyze the data, as well as our collaborators on the data I'm going to talk about today, Eric Marsh and Oren Davinsky. Jose Dupuy is a statistical geneticist at Boston University, and she helped on some of the analysis as well. And Kevin, who initiated the project. So I'm going to have two parts to this talk. One, investigation of genetic factors that influence response to cannabidiol for treatment of epilepsy. And this is actually looking at the same group of patients that you just heard about from Elizabeth and Oren in this open label trial. And so some of the things that we may have found there are some response differences in CDKL5 patients, patients with mutations in that gene, and then some regulatory SNPs in another gene that may be associated with better response. But it's still early days. So, and then the second part, I'm going to talk about rare variation in endocannabinoid system genes. And here we've found rare variants in the cannabinoid receptor type 1 and diacylglycerol lipase, which are associated with neurological phenotypes. So early on, we got involved with folks running this trial, Oren and Eric Marsh, and more recently we brought on other collaborators who are also participating in this trial, the open label trial. And so what we've been doing is essentially analyzing the DNA of the patients who are in the trial and looking for factors that are involved with response. And what I'll tell you about today are the results from the first 75 patients that we've analyzed. Cortigen, our main sort of thing is neurological testing for neurological disorders, epilepsy and seizure disorders, neurodevelopmental disorders, autism, mitochondrial and functional disorders, a whole series of different gene panels, comprehensive panels with large numbers of genes, and actually whole exome sequencing as well. So the panel we used for this study is called EpiSeq Comprehensive. It has 471 genes. It covers the major epilepsy-associated genes, GABA pathway genes. We included in the panel from very early on the major endocannabinoid system genes, so the CB1, CB2 receptor, Degla, and MAG lipase. And then it also contains a number of genes that are actually associated with endocannabinoid system response. So this was more or less by accident, but when we actually went through and analyzed a lot of data from a large number of genes, we found that a list of genes there that when patients are either given CB1 agonists or various other compounds, that the activity of these genes or their expression changes. And it also includes genes involved in drug metabolism. So basically, we're looking for a genotype-phenotype correlation. So significant variants are identified, reviewed, and verified. The clinical data is considered in relation to the patient's disease and the known effects of mutations in various genes. And then we basically score the variants on a 5-point scale, so either from benign to pathogenic. And in many cases, we're able to identify what we call a driver variant, which is simply a rare variant in a particular gene that explains the patient's disease and phenotype. And so from this study, the 75 patients, we identified those types of driver variants in about little over 40% of the patients, 32 patients. So, okay. And this actually shows kind of a distribution of those driver variants from that we identified in these patients with the response curve. So each bar there is a patient, and it's the same kind of response diagram that Elizabeth showed you. So we've got, the good responders are down here, and the poor responders are up here. And when we actually look at the genes across that spectrum of response, we see SCN1A it pretty much covers the whole range of responders. Even though we know there's a good response in Dravet patients, which have these SCN1A mutations, there's no actual statistical correlation with the presence of an SCN1A mutation and response. On the other hand, with CDKL5, they kind of stack up towards the poor responding end of the spectrum, and there are no really great responders that have CDKL5 mutations. It's not reaching significance here. It's still above 0.05. So it's a possible indication, but and we need more data to really investigate this further. But that's where we are right now. So CDKL5 is interesting. It's a protein kinase involved in transcriptional modulation of synaptic plasticity. The process is dependent on NMDAR, the NMDAR system, and there are other genes in that pathway that we know also are associated with refractile seizures. So we're not sure. CBD just may not influence this pathway as strongly as other pathways, but obviously need more data to look into that. So the next thing I'll move to is drug-drug interactions. You heard already that clobazam, and this is a paper actually by Elizabeth Thiel's group, has an influence, and that there's an interaction between clobazam and cannabidiol, which leads to increase of this metabolite, N-desmethylclobazam. We looked across these patients, all 75 patients, and here you can see them sort of sorted by response again. So the good responders are down here, and the poor responders are up there. And we actually looked at the data that was available for clobazam levels, which are the green dots across the middle, and N-desmethylclobazam, which are the blue dots here. There's limited data, so not all the data is complete for these patients. So what we do see, and this is reported in the paper, there's most of the patients down at the good responding end happen to also be taking clobazam. So there is clearly an interaction there that's significant. On the other hand, if you actually look at the N-desmethylclobazam levels, there's no correlation with the response relative to the level. So it's not just simply a level of N-desmethylclobazam or clobazam itself. We also looked at the drug metabolism variants. From this data set, we don't see anything popping out in terms of that any one of these enzymes which influence the metabolism of CBD is associated with a good response or a poor response. But again, we have limited data here, so this is early days from that. So this made me think, I mean, clobazam and CBD, perhaps they act on the same pathway. Clobazam's a 1,5-benzodiazepine. It targets the GABA receptor, particularly the alpha-2, ones with alpha-2 subunits. There are a number of GABA signaling genes involved in various epilepsy syndromes. And so, this is interesting. Perhaps CBD acts on this pathway. And in fact, there's a recent publication that just came out in the Journal of Pharmacological Research by Bacchus et al. That shows that CBD is actually an allosteric agonist of the GABA receptor and also prefers these subtypes that have the alpha-2 subunits. So this is an interesting finding, and perhaps these things are both kind of sort of working in an additive way in patients that are receiving both. So I'm going to shift and talk about common genetic variants now. So far these have all been about rare variants that we've been seeing. So you can take an approach of genome-wide association studies to study common variation and its association with disease. The CBD study sample size is too small for this. So what we are doing is using the data, using the data from the oops, sorry. Yeah. Sorry. Using the data from our gene panel and essentially taking the common variants which are nearby the exons of known genes and looking there for a signal that might be associated with response. And so we took, we recalled all the data, so we had consistent variant calls on the whole data set, filtered the variants for minor allele count and Hardy-Weinberg compliance, and then basically look for the allelic distribution of individual variants and essentially testing for correlation with response in that distribution of variants. So an initial pass at this back in 2015, looking at the entire panel, it was about 6,000 variants, we immediately realized that we had too many variants to reach a decent significance level for this data set. But just looking at a very small fraction of the data, we noticed that some of these genes that we're seeing there are actually genes that have some association with endocannabinoid system and the response of the endocannabinoid system. So what we decided to do was focus in on just the genes. There are 39 genes in the panel that are associated with the endocannabinoid system, as I described earlier, either bind cannabinoids or have activity changes associated with them. Those genes are only about 8% of the panel, but 3 out of 4 of the top genes that we saw in this initial pass were in that category. So we thought that was significant, and we should just focus in on those. And so we repeated that analysis on the 75 patients. Here we're seeing one particular gene is approaching the significance level that we would like to see. Still not enough data or enough patient samples to give us a significance level, but it is approaching it. This gene is interesting. It's glial fibrillary acidic protein, which is involved in differentiation of astrocytes. And essentially, the CB1 and CB2 blockade results in persistent overexpression of this gene, and anandamide promotes neural precursor cell differentiation into GFA-positive astroglial cells. These variants that we're seeing, there's 11% minor allele frequency. They're in a regulatory region that has promoter elements, enhancer elements, and actually changes messenger RNA levels for a few different genes. So there's something going on there that looks interesting. But again, we haven't really reached the significance level to say that this is actually a response element at this point. So I'm going to switch and talk about the second part of the talk. This is really a separate study looking at the core ECS genes in our panel. And it's not just the EpiSeq panel, but also our so-called NucSeq panel that looks at mitochondrial disease has these genes included, or a number of these genes included in it. So we have the CNR1, CNR2, those are the two major receptors, FA, which breaks down anandamide, MAG lipase, and diacylglycerol lipase alpha, which produces 2-AG. So basically what we're doing is looking for rare variants in these genes in a large data set. So since these genes were included in these panels, and we've been basically sequencing patients and delivering tests for several years now on these same panels, we have large numbers of samples. So over 3,800 EpiSeq tests and 2,100 NucSeq tests that we're looking at. And so basically what we did is just parse out all the phenotypic terms for all the cases where these variants were observed, select terms that were present in at least 20% of those cases, and then if there were some rare variants shared amongst those, we included those, select a bunch of random controls that do not contain those variants, and just perform a Fisher exact test to test for the phenotypes present or absent in cases versus controls. When we did this, we did not see anything significant for CNR2, FA, or MAGL, but we did for CNR1 and Degla, and that's what I'm going to tell you about. So here, for instance, in CNR1, we found 24 cases that had rare variants, rare potentially deleterious variants in this gene, and looking at cases versus controls for migraine and headache, we see significant association of the 4.5-fold increase of migraine headache. Same for sleep disorder, larger increase associated with those rare variants. Anxiety did not reach significance by itself, but in combination with migraine or headache or sleep disorders was very significant and a significant increase in risk. So, so we think, there are people walking around out there who have these kind of rare variants in their CNR1 gene, and this could be an explanation for part of this disorder. And actually, so interestingly, there were 2 out of 6 cases that had one particular variant that shared the same variant, and they have combined sleep disturbance, anxiety, and abnormality of the autonomous nervous system, completely unrelated patients with similar phenotypes. So when you knock out the CB1 gene in mice, they display behaviors of elevated avoidance, freezing, risk assessment type behaviors. They have an early, accelerated early learning as well as early memory decline. Those are all the phenotypes you actually see when you knock out the gene. Oops, sorry. And there's also this thing called clinical endocannabinoid deficiency, which many of you have heard about, theory promoted by Ethan Russo for many years. It posits that disorders like migraine, fibromyalgia, and irritable bowel syndrome are related manifestations of low endocannabinoid tone. And these things, these frequently occur together, and they respond well to THC. So what we're seeing in these rare variant cases seems to parallel that, and so it kind of suggests that these patients would be good candidates for CB1 agonist therapy. So given that there is a crystal structure of cannabinoid receptor, we decided to just take a look and see where the variants map on that structure. So it turns out they actually map to the 7 transmembrane domain near the binding pocket or around that domain, relatively few in the cytoplasmic signaling domain. So we think that's interesting. Perhaps this domain is more tolerant to variation in humans. So for diacylglycerol lipase, we see a similar thing, different phenotypes. So here we're seeing an excess of seizures in these 36 cases that had rare variants, excess of developmental disorders. One particular rare phenotype simply labeled as abnormality of brain morphology. We see that increased at significant levels. And then combinations of these. And these are all quite significant. So it looks like we have an associated with increased susceptibility to seizures and developmental disorders. These are enriched in our database by about 7 times compared to NOMAD, which is actually a database of ostensibly normal people, about 130,000 individuals. So, significant enrichment in a disease population and a number with 0 frequency in NOMAD. Again, we see some shared phenotypes. So this particular rare abnormality of brain morphology is highly enriched in Degla variants, including 2 of 5 patients with one particular variant that they share. So there's no crystal structure for Degla, but you can see that these variants kind of cluster in the 3' end of the protein, or at least of the secondary structure. This particular domain has a lot of phosphorylation sites and is probably more tolerant of variation. So I won't go into this, but essentially Degla is involved in neuronal development. There are a number of studies in knockout mice that show it alters nerve development in different parts of the brain when it's knocked out. And so we think this is interesting and significant, and it just definitely warrants additional study on other endocannabinoid system genes. So basically, we found these rare variants. We have some putative associations from the CBD response study, and we're basically working now on following up with some additional patients from Elizabeth Thiel's group and Jerzy Slofarski's group, about 144 additional patients, plus Eric Marsh, that we should be able to push these associations over the top, hopefully. And I'm just going to end on a quick note. We're always seeking new collaborations for these kind of genetic studies, try to understand from the genetic side what's going on when we treat patients with cannabinoids. We've been developing some new resources, panels to look at pain, syndromes, migraine, abdominal fibromyalgia, a whole bunch of genes that we basically pulled in so that we can use this in a study to identify additional factors that might be involved in response. And Cortigen has a new subsidiary called Avantra Genetics, which you may be interested in. We have panels for migraine, abdominal pain, autoinflammatory disease, and other conditions. And so you can actually talk to the folks out at the booth, and they might give you some helpful advice.